Okay, good morning, ladies and gentlemen. This webinar is brought to you by the Institute of Advanced Studies. My name is Lee Suing. I'm the director of the Institute, and I will be your host today. Today, we have 500 people who have registered from the NTU and NIE community and alumni, as well as friends of NTU. And they have all joined in the webinar. And the hot topic of today's webinar is COVID-19, science, medicine, communication, and education. So let me first introduce you to the storyline and the expert panel. So on the top, we have the bogeyman, which is the coronavirus shown at the top. And today we can see this tiny coronavirus with a scanning electron microscope, or SEM. And there are quite a number of SEMs on our NTU campus. Well, how tiny is this virus? It has a diameter of about 0.1 micrometer or 0.1 micron. But how small is it? Well, if you take a printed full stop, which is something that we can see, we can barely see, the full stop that you see on the typewriter is about 0.3 millimeters in diameter. And then knowing the dimensions, I'm sure we can show that we can pack in about 5 million coronaviruses in one layer on a tiny full stop. So you can imagine how tiny the coronavirus is. So our storyline will take us from science to medicine, to communication, and finally to how education on campus has to adapt to the pandemic. We have 4 distinguished members of the panel to speak on each of those areas. And our members of the panel are Professor Jo Yoon, Professor David Lai, Professor Mei Luin, and Professor Kam. And I will introduce them before they make their presentations. Now, just some housekeeping instructions from my production team. So we will disable the raise hand and chat features for the audience. We will raise questions via the Q&A button at the bottom of your device screen. So before I call on Joe, let's take a pause and a short quiz, first pop quiz that you can vote on so that we know we are on the same page. So it's very surprising. One estimate says that each of us harbors about 400 trillion viruses. So it is in between those unimaginable huge numbers up there, between trillions and quadrillions. So most of you got the answer correct, I would say. And fortunately, most of them, these viruses can coexist peacefully with us. But from time to time, however, we may encounter alien viruses for which we have no immunity, like in the case of COVID-19 today. So thank you for taking part in this poll. What I'm going to do now is to introduce our first speaker, Professor Yoon, Professor Jo Yoon. Now, Jo is the Research Director for Biomedical Sciences in the President's Office, and he's from the School of Biological Sciences. Jo's student, Jonathan Gunn, has just completed his PhD thesis on the discovery of small molecule therapeutics for the MERS coronavirus, which is related to our COVID-19 coronavirus today. So today, Jo will talk about the science of coronaviruses. So Jo, over to you.
Hi, good morning, everybody. Thank you for joining our today's webinar. You can now see my screen, I guess. So as COVID-19 became a global pandemic, every nation, public health authority, stakeholders are trying their utmost effort to combat viral transmissions. Although we are living in an unprecedented time on the way to post-COVID-19 world, in our response to the pandemic crisis, we are also witnessing one of the greatest moments in history of science. So we just got to know new coronavirus only a couple of months ago. Started publishing this coronavirus like January or end of December. But our knowledge about the previous two outbreaks, namely SARS, one in 2002, and MERS in 2012. So those findings, outbreaks, help us to quickly understand the nature of virus and the virus-mediated disease, COVID-19 or SARS or MERS. So in this direction, today I'll briefly touch upon general feature of coronavirus and their life cycle and their famous cellular receptor H2 biology, then our body's immune response, and finally, like briefly touch upon the current status in therapeutics and vaccine development. So coronavirus, there are hundreds of coronavirus, mainly in animals, but taxonomy structure reveals that they belong to nidovirales. So they are positive sense, single-stranded RNA virus. There are different types of viral genomes, double-stranded, single-stranded DNA or RNA. The coronavirus is enveloped positive sense, a single-stranded RNA virus, just like a dengue virus or hepatitis C virus. They are all same positive sense, single-stranded RNA virus. The size is about 100 to 150 nanometer in diameter. One of the most interesting features of the coronavirus is spike protein here is projected from the virion surface, making like a corona shape, so as the name reflected. So we already did the polling. We are living with the bacterial virus. Human microbiome or virome is, we are quite familiar with this development in recent years. More than trillion or 100 trillion microbes coexist in our human body. They can get in our body when we breathe and we touch object or surface. However, most of them are harmless, but some can make us sick. First human coronavirus was reported 1965, nearly 60 years ago. Until today, 7 human coronavirus have been identified as shown here in this table. Although they account for 10–15% of common colds, but they receive little attention due to their mild phenotype in the human body. But this perception has changed with SARS outbreak in 2002, Guangdong, China, and MERS outbreak 2012, Arabian Peninsula, and the recent COVID-19, Wuhan as well. So because it causes severe lung damage or respiratory syndrome. A lot of genetic analysis indicate that the coronavirus is originated from bats. But still question remains whether the virus traveled directly from bat to humans or there's intermediate host involved. Jury still is out there, but of course there are many findings suggest there are intermediate hosts involved. But in COVID-19 new coronavirus case, still we have to wait for further study to answer the questions. SARS coronavirus 2 contained a large RNA genome. It's about 30,000 nucleotide long. It uses spike protein to get into cells, and then it releases genomic RNA, is positive single-stranded. And then they're making two large open reading frame, 1A, 1B. So open reading frame means they can be translated to make proteins. So basically these two large open reading frame to make two polyproteins, PP1A and PP1B. But sometimes there is a frame shift, so you make even larger protein, PP1AB, together. Regardless, this polyprotein is further processed by virus protease, and then they are making part of replication transcription complex. They are making first a negative copy of its own genomic RNA, and then also is make copy of its own positive sense RNA. In parallel, using the negative sense RNA, they are making subgenomic RNA to translate other viral proteins, which is basic structural proteins, together with this smaller structural protein with the genomic positive sense RNA. They are assembled. And then they are hijacking human, the cell's machinery, then they are trafficking and they release out of body. That's basically how the coronavirus they work and replicate and transmit and infect other cells. Now we'd like to discuss a bit of the receptor stuff. This is angiotensin converting enzyme 2, which both COVID-19 new coronavirus and SARS original one also use the same receptors. When blood pressure drops, the liver releases angiotensinogen, which is an inactive peptide. And then renin is secreted from kidney, convert angiotensinogen to angiotensin 1. And then angiotensin 1 is converted to angiotensin 2 by enzyme, angiotensin converting enzyme. So then we produce this angiotensin 2, which is very important vasoconstrictive peptide, cause blood pressure increase, and then blood volume increase, and then also sodium water retention. Also, if too much, causing the oxidative stress inflammation, also increase the vascular permeabilities. So in response to this increased level of angiotensin 2, the ACE2 kicks in, try to counterbalance the level of angiotensin 2 to change to the smaller peptide, angiotensin 1,7, which basically counterbalance. The problem is when cells get infected by the SARS, somehow, still we don't know how, but ACE2 is rapidly disappear. But that causes a problem because then you cannot counterbalance the level of angiotensin 2, which is, if too much, which is also detrimental to cells. That causes a lot of these problems, especially the lung cells, highly inflamed, and they're flooded with fluid, cytokines, and it impaired oxygen transport from lung alveoli to the bloodstream. Basically, hemoglobin cannot have enough oxygen to deliver to the tissue cells. That makes our breathing difficult. So that causes a lot of disease. So basically, one of the major cause of mortality is acute respiratory distress syndrome. So both SARS-1 and SARS-2 use same receptors, the ACE2. The structural analysis show overall, of course, there is some sequence difference between SARS-2 and SARS-1, but overall topology, three-dimensional structure share almost identical architecture. But if you measure the binding affinity, somehow the SARS-2 bind about 10 or 20 times stronger than the SARS-1. So at this moment, we don't know whether that binding affinity difference can contribute unusually high transmission rate of new coronavirus. Jury's still out there. We need to do a study more to answer those questions. So what about the pathophysiology of coronavirus? In response to the virus infection, our body activates innate immune system, basically activating macrophages, dendritic cells, monocytes, and so on. Basically, they are fighting at the site of infections, while they are also communicating with other immune cells to mobilize a more coordinated defense system to combat the virus attack. So basically, cytokine is essential in maintaining our immune system properly. But for some unknown reason, some people overreact to viral infections. Basically, out of control cytokine response, so-called cytokine storm, can damage cells. So some of the existing drugs targeting either the IL-6 cytokine or their receptors or downstream kinase cascade, the drugs are investigated to whether they can modulate hyperimmune responses. So in response to the COVID-19 pandemic, NTU also community also mobilizing our resources and calibers together to work on a different area of COVID-related research, including drug repurposing, developing therapeutic antibodies, or developing rapid or more sensitive molecular point of care diagnostics. Or we are also trying to produce more engineered antigen and decoy receptors. This is really happening in NTU campus. So when MERS outbreak took place several years ago, one of my students, John, joined my lab and began working on MERS project because there was no approved drug or vaccines available to treat MERS. So he and Hari took a structure-based approach and then he identified a couple of FDA-approved drugs which can bind this receptor binding domain. And then we also came up with one of the asthma drug, which is effective against a live MERS coronavirus. We confirmed in collaboration with the Pasteur Institute Korea in South Korea. So we're hoping that they can be translated for potential human application in the future. So this is my last slide. Scientific and medical community are moving at record-breaking speed to develop treatment for new coronavirus. Some are panning the old molecule, hoping to find effective drugs basically through repurposing approach. But others are applying latest technology to develop more sophisticated treatment of vaccines. There are DNA vaccines, RNA vaccines, protein vaccines, or inactivation of viral particles as well. So we hope that the race will bring result so that the therapeutic vaccines are made available to treat COVID-19. I guess you are going to hear more stuff from Professor Lai on the latest clinical trials. As you can see here, every processes starting from viral entry to the cells or translation or proteolysis or RNA replication, there are all side of potential therapeutic intervention site. So that there are several drugs already being investigated, especially remdesivir, showing quite potent efficacy against this COVID-19. We are going to hear more from David Lai during his talk. So with that, I will conclude my talk here. Thank you very much.
Thank you, Joe. Thank you for the very detailed biochemistry of the SARS-CoV-2 virus. and I'm sure the students in the audience are enlightened by your presentation. But for the students in the audience, some of the content may be over your head. Well, it is for me, because some of the biochemistry I do not quite understand. But do not worry. I remember that the Nobel laureate, C.N. Young, and we have a C.N. Young program, scholars program on campus. And C.N. Young advised that that is normal because he had the same experience when he attended seminars when he was young. Most of the time, he never understood most of the material that was presented. He said that we learn by osmosis, and later we will go out and find out about things that we do not know. So that's the benefit of attending seminars. So before we move on to Professor David Lai's presentation on the medical science aspect, let's have a short pause and take a second pop quiz. So here we go. I think most of you felt that there are no effective treatments at the moment for COVID-19, but it is also clear that quite a number of you are following the news very closely, where there's a lot of reports on HIV drugs and the Ebola drug remdesivir used for the treatment of COVID-19. So you will soon learn about the answers from David. So with that, let me introduce Professor David Lai. David is with the Lee Kong Chian School of Medicine, NTU, as well as the Yong Lu Lin School of Medicine at NUS. So he's a senior consultant in the Department of Infectious Diseases at Tan Tock Seng Hospital, and also the director of the Infectious Disease Research and Training Office at the National Center for Infectious Diseases. David is also the president of the College of Physicians in Singapore. So David has been on the battlefield of the COVID-19 outbreak in Singapore. So he will now give us his talk on evidence-based update on the treatment of COVID-19. David, please.
Thank you. Can you guys hear me? Yes.
Yeah.
Good. And so my primary appointment is actually Director of Research in the National Center of ID. If you're unlucky enough to have COVID, you may have been housed there for a few weeks, and it's a very nice place. And I've joined appointment with the Lee Kong Chian School of Medicine. I'll try to give you a very quick but evidence-based update of what is the effective treatment for COVID-19. So this is an unprecedented epidemic. Now, a pandemic. This was published in April, and at that point they counted more than 300 clinical trials. But for treatment trials, there were more than 200. There are probably more now, but only 157 are randomized controlled trials. You cannot tell if a treatment will actually work unless you test it in a randomized controlled trial. Otherwise, it's just observational. Any effect may just be chance. Now, of all these drugs, you can see a lot of people tested Kaletra, the HIV drug. A lot of people tested the malaria drugs, hydroxychloroquine, and so on. And there were a few trials on stem cells, a few trials on antibody, and but also 10 trials on remdesivir. So most of these trials, even among the randomized control trials, a lot of them do not actually have a control group and definitely not a placebo group. So most of these trials, in fact, will not be able to inform on the final evidence. That's number one. Number two is that we look at this publication. Most of these trials have fewer than 100 patients. That tells you these are not a phase 3 trial, and they will not actually provide the answer. It's very unfortunate that there's a rush to do trials, but not good quality trials. And you can see that a lot of these trials initially started from China. It's a lost opportunity, and subsequently US and Europe comes in, and now they are mostly in the Europe and US, but also internationally. So if you look at this life cycle of the virus, as the virus attaches itself to the host cell membrane, there's a potential target, which is the There's no target for ACE2 receptor yet, although there is a recombinant ACE2 being trialed as well. But one of the purpose drug is camostat or mafemostat inhibits this transmembrane protease serine 2 that is available in Japan and Korea. And then once the virus enters the body, there is this flu drug that's only available in China and Russia called umifenavir, which potentially can work. And this is also where the malaria drug hydroxychloroquine works by inhibiting viral entry and endocytosis. And once that happens, then the protease, the Kaletra drug is a protease and it works at the proteolysis step. But if the virus gets through that, then remdesivir and ribavirin and favipiravir is an antiviral that is working hard in the last 20 years to find a virus to treat. That is where remdesivir works. And then once the virus gets out, then it triggers inflammation, and that is where anti-inflammatory treatment is supposed to work. So remdesivir. Now, at the start of the pandemic, we were actually reviewing all these. We know that any new drug will not be able to get to the patients in time because of the process of preclinical animal phase 1, phase 2 study. So we were focusing on repurposed drug. And of all the repurposed drug, the only ones that really have a glimpse of hope is the HIV drug Kaletra. And we did not think the malaria drug will work because it has failed in influenza, it has failed in dengue, in randomized controlled trial. And then there's this drug remdesivir, which is actually itself a failed Ebola drug. So this study came out of Wuhan. It's a state laboratory. It's a very good laboratory on virology. And I point to you here, remdesivir. Its EC50, effect concentration to inhibit virus, is 0.77. It's one of the most potent. You can see that chloroquine is 1.13. If you look at favipiravir, the antiviral that's looking for a virus to treat for the last 20 years, is about 60. Look at ribavirin, which was used to treat SARS and MERS, is over 100. You look at the pancreatitis drug in Japan and Korea, the EC is about 20. So it's one of the most potent drugs that we have. And in fact, NIAID and Gilead quickly did an animal study which shows that animal treated with remdesivir did not show lung infection. In fact, it could reduce the virus in the lung. And then when they sacrificed the monkey, there was also less damage to the lung. So there are a few key trials of remdesivir worldwide. So Gilead itself ran 2 trials, the severe trial and the moderate trial. Now, the Gilead trials are not meant to answer the question whether it is effective. It's mainly trying to see whether it can use it for 5 days rather than 10 days in Ebola, because if that is the case, it can actually extend the stock to treat double the number of patients. And as you can see, both studies had more than 170 sites across US, Europe, Asia, and Singapore participate in all 2 of them. In fact, Singapore General, National Hospital, and NCID are both trial sites. You can see here, obviously, US recruited the most patients, followed by Italy and Spain, Switzerland, and Singapore is really doing quite well as well over there. We have recruited, actually, as of yesterday, we have recruited 98 patients in remdesivir trials in Singapore. And this was an announcement 2 weeks ago, essentially comparing these in severe COVID. These are patients who require oxygen, 5 days versus 10 days. You can see that whether it's clinical recovery, discharge, or death, absolutely no differences. And in terms of side effect as well, presumably if you take for more, you may, but actually there's no difference as well. So therefore, a 5-day course as effective as 10 days course, and the current limit supply of 140,000 courses can treat double the number of patients. So this manuscript has been submitted to New England for review. Now, the NIAID trial, the NIH ACTT trial, this is called Adaptive COVID Treatment Trials. This is actually the definitive trial because it compares remdesivir versus placebo, and it completed recruitment 1,000 patients in 58 days and 68 sites, and Singapore is also one of the sites. We are now trying to start the second trial comparing baricitinib versus placebo. This is a JAK inhibitor, but remdesivir is going to be the backbone. Now, so there was some controversy on the 29th of April because the director of NIH, Dr. Anthony Fauci, announced that in this trial, we found that the patients recover faster. That's significant, 30% faster. What do I mean by that? So in the placebo arm, they took 15 days to recover. In the remdesivir arm, it took 11 days. But there was also a hint of a survival benefit. And what do I mean by that? On the placebo arm, 11.6% died, and on the remdesivir arm, 8% died. So that's about 30% reduction as well. And the p-value is just boring significance. Now, what was a controversy? In general, we don't announce results like that without the full analysis and peer review. The reason for the announcement was because after the Data Safety Monitoring Board, this became obvious, and there were still patients in the trial on the placebo arm who are continuing. They could be not doing well, and we felt that there was a medical obligation to unblind and offer them remdesivir. So this is the main reason why it was unblinded, and it has nothing to do with Gilead. It was a decision taken by the investigators, including myself. Now, there was at the same time, on the same day itself, the China remdesivir trial was also published, and this is the one that the WHO leaked the results a week before and says it did not work. Now, what is this thing behind this? If you look at this, the placebo recovery is 23 days, and the remdesivir is 21 days. And look at the mortality. Early treatment is 15% death in the placebo, is 11% death in the remdesivir. Now, this trial was stopped early. It was stopped early because China could no longer recruit patients because the pandemic was over in China. And they needed 453 patients, they recruited 237. That's number one. Number two, because of the early termination, it was never powered to show effect. Now, it would be very nice if it showed effect, but it wasn't powered. The other problem about this study is 2-to-1 randomization. You can see for every 2 patients remdesivir, it needed 1 placebo, so it's underpowered to show anything. But nevertheless, it just shows the importance of doing properly powered trials. And the direction of benefit is towards remdesivir. I'll now go to Calitra. We call it Calitra. It's actually a 2-in-1 tablet. It's not a combination, lopinavir, ritonavir. So this was one of the first randomized control trial published out of China. It started recruiting in early February. It completed recruitment and was published in middle of March. You know, in peacetime, you could not finish a randomized control trial in anything less than a few months, but in this pandemic, it is very different. The Gilead Severe trial finished recruitment in 4 weeks. This trial finished recruitment in 4 weeks, and the Nanyat remdesivir trial finished recruitment in 2 months. So in this particular trial, it shows that there's no difference in improvement, 16 days, mortality, 25 to 19%, clinical improvement, likewise, not much difference. The statistical event is not shown for anything. Now, is that surprising? As I told you, we weren't particularly optimistic about this drug working. So, and there's some controversy about this study as well. If you look at the New England paper on the 13th of March, it says that in early treatment within 12 days, there was shorter time to clinical improvement. But on the 14th of April, if you look at it again, it says was not associated with short time to clinical improvement. There was no announcement, but and people that read the first paper still think that early treatment may work, but in truth is that after some tidying up of preliminary data, there was no benefit. Now, it's just surprising. This Nature Review Drug Discovery paper explained it. The HIV protease is a aspartic protease, and the HIV protease inhibitor is designed to fit into that. Whereas coronaviruses, they are cysteine protease, so they're a bit different. So you don't really expect it to have high binding affinity and to be truly highly effective. Is that the end of the story for Calitra? Now, this came out last weekend. This is a study done in Hong Kong. It's a phase 2 trial. Phase 2 trial primarily is not designed to answer the question, is the treatment effective? It's designed to answer a question, is the treatment safe in patients? So you can see it's not a lot of patients, only about 120 patients, once again, 2-to-1. And they gave Kaletra with ribavirin, which is a hepatitis C drug, which causes a lot of side effects, and interferon beta 1b by subcutaneous injection under the skin. And what did it show? It shows faster RNA clearance and hospital discharge. Now, we keep the patient in the hospital until the RNA is cleared, so these two are linked. They also show faster symptom recovery. But these are actually the sort of, if you actually look at this trial, these are the same sort of patients we send to Expo, we send to NTU Resort. So they have very mild infection, which don't really need treatment. So it is a very interesting study. It shows that this triple combination is effective in mild COVID-19. Kaletra on its own is probably not going to work in serious COVID-19. But in mild COVID-19, if you give 3 drugs, including an injection, it may work in patients who will recover on their own anyway, just a bit faster. So that is the conclusion of this trial. I will now come on to the malaria drug, okay? They will never, one of the reasons why this was tested was because remdesivir supply is really, really limited. So as the pandemic exploded in North America and in Europe, people needed to treat patients. And the only way to do it is to look at whatever you have. Now, so go back to the Wuhan study. Chloroquine EC50 is 1.13. They subsequently compared chloroquine with hydroxychloroquine, and they found that hydroxychloroquine is 4.91, chloroquine is 2.71. So chloroquine is supposed to be more effective than hydroxychloroquine. This is the state laboratory from Wuhan. It is a BSL-4, is one of the leading virology lab in the world. There's another group in China that somehow repeated the experiment in a slightly different way and reported that chloroquine's EC50 is now 5.47, and then look and behold, hydroxychloroquine is 0.72. So the proponents of hydroxychloroquine will always quote this study, ignoring the first two, and say that hydroxychloroquine is better. It is safer than chloroquine, but is it better? I really doubt so. Now, and then there's this very, very dubious study from the French professor Didier Raoult, who publicly declared he does not believe in the randomized control trial. There's a blog here that you can read where all the preprint were analyzed to show how dubious the data is. But essentially, in green is the stupendous effect of combining hydroxychloroquine with azithromycin, which is an antibiotic we give for cough and cold. And then in blue is hydroxychloroquine on its own, and in black is no treatment. So this is the study that launched Trump's tweeters, and it's pretty much no one believes it now because there are a lot better studies coming out. I'll show you now. So many French also don't believe him, and so this is from 4 hospitals in France. They look at people on hydroxychloroquine versus nothing, and this is ICU or death, so much better endpoint. This is not about, when we talk about, if you go back to the French professor study, this is about RNA clearance again, okay? We know that RNA can last for a long time, and they don't really differentiate between disease severity, okay? So now this study, bigger, non-randomized, they look at ICU and death, no difference. Severe lung infection, ARDS, no difference. And hydroxychloroquine increased the QT interval by about 10%. So no benefit somehow, but non-randomized. This is the study that came out of China, randomized, but this is probably a phase 2 study, very small, and they show no difference in the RNA clearance by day 28. This is much more credible. This is what we see, observe in Hong Kong, in Singapore, and symptoms, no different. Adverse effects, more, 30% versus 9%. This is a randomized trial. These 2 studies have not been published yet. And then a large study from the US, from the VA hospital, Veteran Affairs, 368 patients. So 97 on hydroxychloroquine, 113 hydroxychloroquine azithromycin, 158 nothing. So essentially it shows that the people on hydroxychloroquine actually had higher mortality. That's because they were sicker, therefore they were treated. But otherwise, you don't really see any benefit here. So this also came out of US. Now, and what is this study? It looks at hydroxychloroquine alone versus combining it with the antibiotic azithromycin, and it looks at your risk of developing abnormal heart rhythm. So if the way we measure is really called QT interval, is basically on your electrocardiogram, how is the timing of your heartbeat. And if this QT interval increase your risk of abnormal heart rhythm. So 90 patients, 53 received azithromycin. This is what a French professor proposed. So now the hydroxychloroquine group actually had a higher QT, and then after treatment, it increased by 5.5 milliseconds. Whereas the group on hydroxychloroquine had a lower QT, and after treatment, it increased by 23 milliseconds, a lot more. So that's a bit of a worry. Another study from out of New York. Now, usually New England does not publish this sort of observational randomized study, but this is obviously a contentious issue. Nearly 1,400 patients, 800 on hydroxychloroquine, and basically no real benefit. Of course, those who are treated with hydroxychloroquine are sicker, so unadjusted, the risk of death is 30% versus 15%. But no matter how you adjust it, inverse probability weighting with matching with adjusted probability score, there's no intubation or death benefit from using hydroxychloroquine in New York City. So I won't treat anyone with hydroxychloroquine currently. I will now come to this issue of cytokine storm. Now, it's been shown in patients that in a small subgroup, there will be an increase in interleukin-6. Now, just to give you some background, by and large, about 20-30% of patients will be asymptomatic. There'll be studies that shows lower or higher, but on average, 20-30%. Based on our local data, about 40% will not have any pneumonia, so they are very, very mild. Another 20-30% will have pneumonia, but they never need oxygen. Another 20% will need oxygen. So in this sickest group, there's an increase in interleukin-6, which drives inflammation. So this is the first study that came out of China, 21 patients only, and it's non-randomized. They treated tocilizumab, and it shows rapid decrease in C-reactive protein, which is driven by IL-6. IL-6 basically drives fever as well as CRP, and fever rapidly improve, but also a rapid improvement in oxygen you can see in panel D. So this is actually the study of 21 patients only, which led the big pharmaceutical like Roche and Sanofi to launch their interleukin-6 receptor antagonist trials. So far, they are ongoing, and we don't know results yet. This is another study in China with 15 patients. A lot of them are treated with steroid, and I will show you some evidence here. So example here, the C-reactive protein actually improved, but patient died, okay? Here, the interleukin-6 actually got worse. No, actually got better. 1,700 to 700 of patient got worse. So the result is actually quite uncertain, and we really need to have randomized controlled trial to prove whether this will work or not. Now, baricitinib, this is the last anti-inflammatory drug. It is interesting because this was actually discovered by artificial intelligence. So this group from UK, they are from a firm called Benevolent AI, and based on whatever is published out there, based on pharmacology, based on antiviral, based on receptor binding, they deduce that this type of JAK inhibitor will actually work, and the baricitinib will work best. In what way will it do so? So it inhibits JAK1 and 2, which leads to reduce IL-6 downstream. It also inhibits this AAK1, which is for viral entry. So is there any data to show that yet? No. There are patients treated in Italy, there are patients treated in US. The doctors that treated them claim that they work, but at this stage, there's no real evidence yet. And in the ACCT trial that we are going to start on, it will be tested. So this is the end of my talk. I apologize for exceeding the time. Thank you.
Thank you, David. Thank you. Thank you for sharing with us the evidence behind the repurposed drugs for the treatment of COVID-19. And I think it's very useful information for us, because who knows, some of us may catch COVID-19, and we will have to consult you again, and we will know what to expect. So next, before we move on to Professor Maylwin's presentation, let us take a pause and have a third pop quiz. So the median is on some every few days, moderate amount. So these results may be useful for May, who knows, right, for research. And so with that, thank you. And let me now introduce Professor Maylwin. And May holds many appointments. She's the Associate Dean at the College of Humanities, Arts and Social Sciences. She's also the Director of the NTU University Scholars Program. And May is a professor at the Wee School of Communication and Information, as well as a joint professor with the Lee Kong Chian School of Medicine. And then May will now present on observations of COVID-19 infodemic.
Thank you very much, Professor Lee, and welcome to everyone who is joining us this morning. I'm going to be sharing with you observations of the COVID-19 infodemic. And really, I just, for the next 15 minutes or so, I'll be sharing with you about the evolution, the concerns, and the countermeasures in that sequence. So we all know about the prevalence of social media and how much time we spend on our phones and on social media. It was already used in a massive scale before 2019, but the usage has seen considerable increases since COVID-19. For example, if you just look at the Twitter post on COVID-19, the original Twitter post, that alone has exceeded 20 million. So that's been retweeted, that's been shared on so many different types of platforms. So you can imagine that these 20 million posts then went on to create, many, many more millions of information that's come out to you. Who knows? Some of you might be tweeting about our, talk this morning, even as we speak. A report by TechCrunch has shown that WhatsApp has seen a 40% increase in usage, and Facebook, for instance, has also seen large increases. So here is a chart from famous, We Are Social tracking, and what We Are Social does is to track yearly the different types of activities that we do on the media. And it was quite interesting that We Are Social came up with this data in April as a special COVID-19 tracking information. And true enough, they were finding a lot of additional usage on multiple platforms. So the public is looking out for a lot more news. They are watching Netflix and many other types of movies and series. They are using their social media, as I mentioned to you, and they're also spending more time on traditional media. So across all platforms, there's additional usage. And as I mentioned, just imagine sharing across platforms, how one set of information gets shared and then that's reshared and so on. So you see this flood, or what I've shown in the first slide, the tsunami of information that's hitting everyone. So now let me talk a little bit about the term infodemic and in the context of COVID-19. Now, infodemic and infodemiology has been something that's been recognized for a number of years, and Collins Dictionary defines this as excessive amount of information concerning a problem that makes a solution finding difficult. In February 2020, WHO used this to refer to the situation over the abundant information around COVID-19, and not just the abundant information, the fact that much of this information is inaccurate. So, that it really led to a proliferation of scholars recognizing that this was an issue. Many researchers came out to examine this, and on 7th April, the WHO held a roundtable for the scholars around the world who started to work on this area, and and to this day it is really becoming an unprecedented phenomenon that needs to be studied. Now, let me just share with you some information from my previous research. So for about a decade now, my team and I have been looking at the intersection between social media, infectious disease, and how people share information online, especially during outbreaks. So we've done work on dengue and some other types of infectious diseases. But in this particular study that I wanted to share with you, as recently as 2016, when Singapore saw the Zika outbreak, we were finding these types of volume and the movements across the social media platforms. So if you look at the top chart, which has the blue lines, you'll see that the government has put out the information and news on the social media platforms, right? So for example, on Facebook, as the WHO declared the Zika case, and then you've got the first imported case in Singapore on May 13, you see the government tweets down there. And then at the bottom, you see what people are sharing. So in brown, you've got the public's Facebook tweets, the public's, sorry, not Facebook tweets, public's Facebook posts, and the public's tweets. And the top and bottom charts almost see a parallel and similarity, which means that whatever the government or the health authorities and global health authorities like WHO were putting out, this was the information that was being shared by the public amongst each other and across their platforms. So when we look at the data in detail, and you can see our paper here, we actually find that there is a lot of synergy. That means that the information that's put out by the authorities is faithfully shared by the public. Now, fast forward today to COVID, and you see that that is not the case. Not only do we have government and health authorities information that's being shared, but you also have all different types of information that's shared. And just now in the poll, I saw that many of you have said that you've recognized that you were exposed to different types of false messages and misinformation, right? So, Seiko and Domenico talked about just recently in April. They talked about this particular threat as something that is led by cognitive manipulation. So they talk about the level of infodemic risk increasing with the level of cognitive manipulation that is applied. And so in my area of health communication, one of the ways that we can look at these different types of information is to see how much manipulation there is. And so I've created this chart where you could see right at the bottom the types of unintentional misinformation that goes out. And the unintentional misinformation could be you sharing with somebody that eating garlic is good for you because it helps to boost your immune system, and you do it in a kind way. But somehow when that information is repeated, sometimes taken out of context and shared on multiple platforms, it could move to eating garlic can protect you from COVID-19 and becomes a source of misinformation. And then you've got somewhere in between where you share the content. Maybe you saw it wrongly. Maybe it was something that you had meant to do to warn some people who may not be abiding by safe distancing rules. So you have a message like police conducting checks at residential units to enforce elevated safe distancing measures. And that has some level of manipulation involved. And then we move on to what is now become termed as disinformation. And those are really doctored tweets or posts that are meant to look like official information, but they are not. And so this goes in the category of disinformation. For example, we saw on Channel News Asia that there were a lot of tweets about school closures, along with official types of logos and so on, especially before the circuit breaker period. So you've got that range. And how is that type of information being communicated? Well, what scholars are finding is that there is a very heavy use of private messaging apps. So for example, on your WhatsApp, you're seeing information that's shared with your groups, but then this then gets translated to other types of wider platforms, sometimes to public platforms as well. And then you see this big wave of misinformation happening. Also, the researchers that I've cited here have found that there is adaptation of misinformation to across different languages and across different cultures. So you even find that, you know, when it goes to different countries, the imposter information, right, in terms of the news authorities and so on. So they are kind of matched to the context of the different countries even. So what does that all that mean? You know, and what are the impact of the infodemic? Well, a couple of different types of themes have been surfacing, and one's at the societal level, where scholars have pointed out that, you know, it is likely to reduce the effectiveness of public health messaging because the public is confused, there's ambiguity, and public is also concerned there's unwarranted fear and sometimes panic, and there's a propensity to increase blame and stigmatize groups who they think may be causing certain issues and so on. At the individual level, again, there's confusion at the individual level, and it's been recognized that, you know, the amount of information and sometimes false information can fuel anxiety and other types of negative psychological impacts. And I've got a picture, of course, from the famous hoarding event that was not just in Singapore, but in so many other countries, that was possibly led by much of the misinformation that floated around. So let me share with you what I meant about, you know, the emotions, the misinformation, and the type of sentiments. In a recent study that we've just concluded in partnership with A-Star, you see that we tracked these over 20 million original tweets across the world. And we started on 28th of January. If you remember, that was Chinese New Year. And when, you know, this was just breaking in Singapore, and you see the green line where we were able to detect a lot of components of fear. So people were very fearful. You know, they were worried, they didn't know what was happening. And all the other types of emotions were pretty much pretty low. The red, anger, you know, the blue, sadness and joy. And over time, you see that the fear component has really come down. And at a certain point in March, it almost matched the components of anger. People across the world have become a lot more angry, and this is fueling a lot of potential negative societal trends. It was heartening to see that we also saw the rise of what we term joy, which is actually a psychological construct, but it is made up of many components like resilience, positivity, and support, right? Especially for the essential workers. So that was good to see the yellow line coming up as well. So what have, you know, scholars learned about countering false information? Some of this info, you know, I've got from various types of literature looking at crisis communication. And we know that these are the 4 different groups that are instrumental, especially during health crisis: the government authorities, whom you would have seen undertaking regular briefings, putting up information on government websites and so on. The medical and scientific community, like my 2 previous speakers, who provide expert recommendations. And sometimes, you know, doctors do it even on a one-to-one basis, right? When somebody does e-telehealth consultation and so on, and they're worried, the doctors are the ones who are able to share the scientific information. The traditional media organizations, in the past, they would have taken different types of news and they would have shared it, but now they really have to tighten the fact checking of the news and what they are putting out. And then all different types of communication companies, which include social media platforms, telecoms, which need to really track what's happening in terms of the misinformation, disinformation, and so on. I think I've just read that Facebook recently is going to be putting up, or they're going to flag the episodes of disinformation. So that's heartening. There are also other types of initiatives to counter such information. Here, Social Media Lab from Ryerson University created this dashboard which tracks and visualizes debunked coronavirus claims by trusted professional fact checkers around the world. And what was interesting is that they even include information that's not in English with an auto translation device. So this is something that the scholarly community can go and utilize. But most important, countering fake information and fake news is really not just the domain of the experts and the various organizations and the government that we saw earlier. It is in the domain of every single one of us. Julie Brennard on 7th April, she talked about the importance of education, and education as the necessary tool to what she calls immunize, providing the type of digital literacy that we learn, but around the particular health episodes to understand and shield against fake news. And she called for the better provision of counter information that's equally better evidence or more persuasive, especially from the scientific community. And I kind of went back to a Schmidt and Bench study. They were doing the study in the science rebuttal context in 2019, and he had done very interesting experiments where he included rebuttals into these fake information, and basically he found that Schmidt and Bench found that the rebuttal approaches mitigate the damage of the science deniers, and even in a small form, rebuttal is effective and even effective in vulnerable groups. So, combination of what those two calls really, led me to believe that us in the NTU community with our 30,000+ group of social media users, and places like NTU and other universities, we've got the resources oftentimes to provide rebuttals and the scientific information that we can be proactive too, especially when we see disinformation and other types of news, to go out into the social media space and provide scientific evidence for rebuttals. And if you're interested in this, in fact, Schmidt and Bench had provided different types of solutions or strategies, like, for example, countering false logic and then debunking the so-called experts and so on. So everyone, I believe, has a role to play. And for my last slide, I'd just like to share with you the study that we are leading at the Wee Kim Wee School of Communication and Information on the influence of mainstream and social media and public preparedness during a pandemic. And really, we are looking at all these COVID-19 sentiments on social media, as I've shared with you. We're also looking at the mainstream media coverage of COVID-19 crisis and misinformation. We're looking at Singapore, but we are in a global consortia that's sharing this information across different universities. And then we're looking at the impact of all this information on Singaporeans' attitudes and behavior responses via different types of studies. So I'd just like to end here, and I'd like to thank my team and all the wonderful collaborators across Singapore and across the world. Thank you very much. And also I'd like to recognize the funding agencies, of course. And thank you very much for listening.
Thank you, May. Thank you. Thank you for sharing with us so much information about the infodemic. And I'm sure that we will try to behave and propagate only evidence-based information and hopefully to reduce the infodemic. So before we move to Professor Kam's presentation, let us have another pop quiz. We take a pause. All right. So most of you would like to see a balance of face-to-face and online teaching and learning. So thank you very much for participating in the poll. And I'm sure the results would be useful to Prof. Kam as he executes the teaching and learning strategies for NTU. So with that, let me introduce Professor Kam Chan Hin. He really needs no introduction. Professor Kam is the Deputy Provost for Education in NTU, and he is from the School of Electrical and Electronic Engineering. So he will share with us adapting university teaching and learning to the COVID-19 pandemic. Professor Kam?
Thank you very much. A very good morning to everyone. I can see that we are running a little bit behind schedule, and so I'm going to try to make my talk as short as possible to allow more time for our question and answer session. So I was asked to talk about how NTU has been adapting to the pandemic. Well, the short answer is we have adapted rather well. But I must say that it was with a lot of hard work and effort by a lot of people, the whole university approach, in fact, in making sure that although the pandemic has disrupted so many of our daily lives, that somehow our teaching and learning activities and education must still continue. So basically, I'm going to talk about three things really. Firstly, to share with you what are some of the things we've been doing in adapting to the COVID-19 restrictions. And also looking ahead a little bit, when the circuit breaker will finally be relaxed, what are some of the adjustments that we can expect? And beyond that, what has this pandemic taught us? And I think this is where our digital learning strategy, which we embarked on over the last decade, has in fact proven to be very effective and has helped us to cope with this sudden disruption in our normal activity. So the early measures when the outbreak broke out in February, when things became clear that this pandemic is going to be quite serious, there's going to be transmission and so on. One of the first things, of course, is that in our business, in education, we have classes and we have large classes. So one of the ways to try to reduce the possibility of a lot of transmission, of course, is to try to downsize the classes. So decisions were made with consultation with Ministry of Education and so on, and class sizes were reduced. For class sizes with face-to-face component, it will be reduced to 50 or less. And of course, some of the, this implies, of course, that many of the courses which could in fact go online will be migrated online. And we then need to have a system to support the systems with their online learning. Now, it was for teachers that over the last couple of years, we have actually been working very hard to transit to a cloud-based system. And in fact, we completed the migration as a transition exactly in December, so that when this COVID situation hit us, we were quite well prepared to transit. The other thing I want to say also is that because we have been embarked on this online or e-learning, to complement the face-to-face, the transition by both our faculty and students and staff have been relatively smooth. Okay, so coming back to this point about supporting students with online learning, so that went very smoothly. One of the things that in fact was quite a major problem, of course, was the idea of students going on exchange. Now, there were a group of students who were outbound students who were going to leave towards the end of February and going off to a lot of other countries in the world for the exchange program. So with the situation rapidly deteriorating day by day and the spread and the number of infected and so on, we felt that it was wise for us to then suspend outbound exchange. And I remember there was one incident where literally our team had to go down to the airport on that day itself, where a big group was going to go overseas, and tell them, please don't go, because if you go and if there's a lockdown, you can't even come back. So that was one part. And of course, the other part was we would have lots of students which are on exchange already, who had left earlier and are now overseas. And then the decision was, with health and safety in mind, to quickly work out a plan to recall all the students. So this was done. And in fact, we managed to get all of them back, almost 1,000 of them back towards the end of March, just before our circuit breaker. Now, with the onset of the circuit breaker, obviously, all face-to-face classes have to stop. So lectures, laboratories, and practicum. So that came to a standstill. And as I said, because we were quite well prepared with our e-learning system, all the classes, the assessments, and even the examinations were moved online. In fact, just before that, we were contemplating that, examination being an activity where, hundreds of students would be crowded into an exam hall to take an exam. And if we had to implement social distancing and all the other kinds of restrictions and all that, it's going to prove to be very difficult. We may need more rooms, more spaces. And we have typically every semester 600 to 700 examinations going on. And that's going to cause quite a big headache. So we were planning, in fact, and advising and encouraging our faculty to think about whether there's really a need to have a final exam, and if not, could they, in the interim, try to work out something, replacing it with perhaps continuous assessment, with hand-in assignments, so that there may not be a need to actually have a physical final exam. So the result came back. In fact, more than 3/4 of them said, okay, we will convert. Now, there's just a small group that said that we can't convert. And for that group, which felt that they must go on with the final exam, we then activated e-proctoring system, which to ensure that the exams is carried out in a proper manner. But for lack of time, I wouldn't have time to go into all the details and so on. Another major consideration, of course, is that with all this lockdown and things, coming to a halt, and especially students who were recalled from their exchange program from overseas, that there's a problem that the senior students, meaning those students in the final year, in the final semester or the second last semester. Now, these students may have a problem because their courses which they were supposed to take overseas, perhaps their internships, are now being disrupted. So what do we do about that? So we have then had to decide to allow students to take courses locally for replacement, and we also activated our career and attachment offices to make sure that students who had to do internships can now be given a place in a local company and so on. But of course, that was all done prior to circuit breaker. So when circuit breaker came in, I think that was not doable. Of course, for laboratory where you need to have hands-on, it's really not possible to really replicate that online, although there are some simpler experiments which can be done online, but on a massive scale, that's not done. So the idea then is, the decision then is to defer the lab until the end of circuit breaker. Okay, so we are now in the second phase of our circuit breaker, and looking ahead with things somewhat stabilizing in Singapore, I think we are now looking beyond that and preparing for the easing of the circuit breaker. So what we can expect, of course, is that there's not going to be immediate business as usual. I think we all know that. So we are planning for adopting safe distancing rules, safe entry protocol. All this has got to be made. All these plans have to be made, and they are all working very hard to make sure that this happens. And then there are, we are thinking about how do we, in order to adopt the safe distancing, probably we have to reorganize some classes. So smaller classes, and looking for bigger venues if those in-class sessions have got to be done. But obviously, we are going to leverage a lot also on our e-learning platform so that tutorials, lectures, and seminars could, as far as possible, carry on an e-learning platform. And if we really have to do face-to-face classes, then all those measures, temperatures, and safe tracing and so on has got to be done. And in particular, there are certain large group activities which typically are associated, for example, TOP, or the Transition and Orientation Program, where new students, freshmen come back, they have camps, they stay overnight, they have team bonding activities and all that, all large groups. So all this has got to be rethink in how we can replace that with, without having to mass a lot of students together. And our residential education program, Learning Beyond the Curriculum, which is carried out in all our halls of residence, this has also got to be relooked at to make sure that as far as possible, all large group activities, sporting events, concerts, choir, all kinds of things will have to be redone, and all the other curriculum, extracurricular activities as well. So, but we are quite mindful that, doing things on the e-learning platform is the next best thing, but in some cases, it may not be the best thing. And I think the pop quiz, just before this shows very clearly that many of you feel that, there is a place for e-learning, but it should be balanced with face-to-face. And I'm glad to see that nobody actually chose that, you want only online learning, right? Okay, let me move on. So beyond the COVID-19, as I said, I think we have been, we have been very happy to see that our initiative in TEL, Technology Enhanced Learning, that we embarked on over the last decade, in blending online learning and face-to-face learning has in fact, paid off dividends. Has allowed us to transit into this mode without, a lot of disruption. I think we have put a lot of investment. We have built bespoke facilities. Many of you would know about our specially purpose-built building, the Hive and the Arc. And one of the important things is that, faculty, while they're encouraged to go online and so on, we're not saying that every kind of lesson in every discipline and every course has got to do online. I think our belief is that the blended learning is the way to go. And it is for faculty really to discuss with our learning technology support team to decide how much of a course, whether it's a lot or some aspects of it could be enhanced with technology. And of course, faculty have, we have several programs ongoing to develop faculty in terms of teaching, especially for new faculty that are joining the university. They have a foundation in university learning and teaching program. And increasingly, we are also embarking on creating communities of practice where the professors who are very passionate and doing very well, who are members of our Teaching Excellence Academy, can hold seminars or webinars in our situation now to share best practices and so on. And we are always mindful that our pedagogy should be informed by science of learning. We have a Center for Research and Development in Learning, and we are working very closely with the center to make sure that whatever pedagogical innovations that are being introduced are based on science. And last but not least, with things going digital and going onto the learning, there's a lot of data being generated, and there is a gold mine of data that we can analyze and use it, for example, to determine the effectiveness of certain interventions and so on and so forth, in order to make sure that learning can go on as best as possible. So I think the key thing is that what we are concerned most about is, students must be learning better with any new innovation that we introduce. So I said something about this, that we want to improve engagement and time spent on learning tasks. If we migrate it online, there must be evidence that test scores are improving. And, we must use the opportunity to develop higher order thinking, deeper learning, to allow students to have more time for reflection, critical awareness. Also to use the medium to improve interaction and to have collaborative activities. And finally, also to redesign learning activities in such a way that we promote active learning, where students then take ownership of their own learning. So, anyway, a lot of things could be done, but as I said, we are mindful that you will apply technology in the right way, and that will actually help everyone. So this slide, which is prepared by one of the team in the learning technology, shows that there are a lot of possibilities of improving, the effectiveness of teaching and enhancing learning. And for each course, for each professor, get into a discussion with the learning technology and the pedagogical group, and decide what is the best way to implement your course so that it can improve the teaching and also, most importantly, learning by the students. And I think this is, in fact, an outcome and affirmation that online learning is probably here to stay. and we should then take advantage and make the best use of it. So this is my last slide. Thank you very much for listening.
Thank you, Prof. Kam. In view of the time, we may have about 5 minutes for Q&A, and thank you for sending in your questions. I will try to direct some of them to the speakers. Prof. Kam has shared with us quite extensively on the strategies that will allow us to move forward like a willow bending with the wind so as not to break and as well as to survive during this COVID-19 pandemic. Okay, we have quite a number of questions. So let me pose the first one to Prof. Kam. COVID-19 in a short span of time has impacted every aspect of our life, work, economy, and even politics. So what the new students will choose to study could be very different from the past. Is the university anticipating a shift in enrollment in its courses, perhaps more towards IT, digital-based, and biomedical science-related courses?
Yes, thank you. Thank you for the question. So yes, indeed, we have seen a noticeable trend of applications, especially for courses in IT, in artificial intelligence, data analytics, courses like that. The trend has been happening, in fact, over the last 2-3 years already. Lots and lots of it. I think in terms of the biological sciences, and medicine. Medicine, of course, is a restricted program. So very sure that there are lots and lots of students who want to get in, but due to capacity constraint, we can't take in a lot. So there is interest going into the biological sciences. But I think since the COVID only hit very recently, it's quite difficult to really think. I think post-COVID, in the next year, for example, I'm pretty sure that with the talk that we just heard from our experts from the biological science and from medical school, that there will be attracting quite a bit more of students who might want to get into this field, which is obviously a very, very key area. In fact, for the admission for this year, I think it has not yet closed. Going to be closing soon, and we have not had time to analyze the results yet to see whether there are any discernible trend. But NTU has been very proactive in looking at what are the needs of the economy, of society, and of the job market in Singapore and so on, and also the interests of students pursuing different disciplines and different courses, and have been we have been rolling out quite a large number of programs, and there's a trend also towards launching programs which are combining different disciplines which interact with one another, interdisciplinary program, which has proven to be very popular. Thank you.
Okay, the next question is to Professor May Win. With the pandemic documentary example, do we fall for conspiracy theories and fake news because of the confirmation bias and self-rationalization, or because we genuinely don't know better?
Okay.
May, could you take that question?
Yes. There's a lot of different types of people across the world, right? And how you respond to different types of conspiracy theories or misinformation, it really depends on so many different factors, as well as the group factors, about the group that you may be in, WhatsApp or Facebook, and the types of discourse that's happening on that particular front. So without really doing social science research, it is difficult to understand how people respond and what are some of the drivers and both the cultural as well as in-group, out-group characteristics that occur. And it's likely to be different for different groups. It's important for governments and authorities to understand it in different country contexts and in different communities. So I think this is why, social science, humanities and arts research is really important in the post-COVID world, because there isn't like a one size fits all, right? Even if we talk about, you know, the people who are displaying, as I mentioned, anger and fear, you know, if we assume that everybody is very fearful and keep using kind of traditional messaging that's supposed to tackle fear, but then you've got audiences who are getting increasingly angry, then the responses will be very different. And the way they respond to the different types of misinformation and disinformation and conspiracy theories will be different as well. So I think that there is a need, not just for, you know, science research, but science and medical research, but also for a lot of the research in the humanities, arts, and social science space to address all these issues in the COVID and post-COVID world. Thank you.
Thank you, May. Okay, we have another question. There is a belief that the coronavirus will continue to mutate and evolve into a milder form that will not kill the host. So is this belief sound? And how long will the process take? Well, I believe this has got to do with science. So Joe, would you like to take this question?
Yeah, thank you for the question. I think it's there's several reports came out showing there are some mutations amongst COVID, the new coronavirus. But if you look at the detailed sequence among these receptor binding site, unfortunately, we don't see mutations much. So that means still, I think mutation frequencies are quite low. So that means for virus, they don't feel pressure to change mutation because now it can easily infect a lot of people. So, but as time goes on, there might be more and more people getting immunity, and then if vaccine available, and then maybe the virus may have chance to get mutate. But so far, we don't see that kind of mutation frequency among the viruses. So I don't think for now, I think we have to wait and see.
Okay, thank you, Joe. In view of the time, and there are still quite a number of questions that have come in from the audience, what we will do is that we will answer your questions by typing in from the speakers, okay? So now it's my pleasure to bring this to a close. And first, I would like to thank the speakers, the 4 speakers, Professor Yuen, Professor Kam, Professor Lewin, and Professor David Lai. So, you know, we put together this webinar all in a matter of 2 weeks. So I have to thank them because that's a very short interval of time that we had. And I would like to thank the audience for joining in the webinar. And finally, I should thank the production team that is working behind the scene, particularly Ms. Chris Ong from IAS and Mr. Leonard Sim from CITS. So I think without the production team, we couldn't have produced this webinar today. And of course, the speakers too. You know, you all have done a lot of work and worked very hard as well, you know, to try to trim your presentation down to the allotted time. So thank you very much, and we can have future webinars. Thank you. Bye-bye.
Thank you. Bye-bye.
Bye.